All right, welcome. My name is Garret Guenther with ACEA Biosciences, and thank you guys for joining us today. What I want to show you is first is an introduction to the Novocyte flow cytometer, and then that's going to be followed up by an interesting new RNA flow assay, and then we're going to talk about some cancer immunotherapy applications. So to start out, if you guys haven't heard of ACEA Biosciences, we have headquarters in San Diego. We were founded in 2002, and we have instruments placed worldwide. In San Diego, we have over 30 Ph.D. scientists and engineers that help contribute to the technology. So the technology is really based on two instrument platforms. The first one's called the Accelligence Real-Time Cell Analysis instrument, and that enables us to take real-time measurements of cells without any labels. There's many different models depicted here, and we also have a cardio-specific instrument that really measures a function of cardiac cells. So a couple of years ago, we launched a Novocyte flow cytometer, and we have several different models of that we've launched since then. So I'm going to talk to you about the Novocyte flow cytometer. So just in general, flow cytometry is a really powerful tool to use in research in that we can analyze individual cells. We can analyze many different markers simultaneously on these cells, and we can do this in a quantitative way at thousands of cells per second. So traditionally, flow cytometry has required a skilled or experienced professional to run many of these flow assays, and I want to tell you that ever since I've been working in the flow field for over 10 years. Ever since I started working on the Novocyte flow cytometer, it's the easiest cytometer to use. So what I have here is a depiction of two different instruments in customer sites. You can see that it's a relatively small instrument. It can fit on a lab benchtop. We have an autosampler attached to this particular instrument, and it doesn't take up that much space. And actually, if space is even limited on the lab benchtop, you can see one of our customers put the computer on top of the instrument to really conserve space. So as I showed, we have this pictured here with the autosampler attached. We also have the standard single tube loader as shown here. So when you open up the instrument, this is kind of what the optical bench looks like. So up at the top right-hand corner, we have our lasers. We use high-quality lasers. They're made by Coherent. They're the Obis line of lasers. So they're very reputable. They're very stable. They have a long lifespan. These lasers here are pointed down towards the flow cell, where they can then integrate the cells or particles of interest. So for our sample acquisition, we use a syringe pump that drives this, and this allows us to get precise volumetric measurements, which equates to absolute cell counts in every single sample. So once the lasers excite our markers of interest, the light is then collected, they're passed through different filters and collected in our PMTs off to the left. So over here is our filters. They're made by Semrock. We have our PMTs made by Hamamatsu, so these are all high quality components. The interesting feature about our detection system is that we have a 7 log dynamic range, and this really means that we have a very large window of detection, which negates the need for any PMT voltage adjustments. So in a traditional flow cytometer, you usually have like a 4 log or 5 log dynamic range. So you have a more narrow window of detection compared to the Novocyte. And so this narrow window of detection means that you have to adjust your signal so it falls within that narrow window. So up here at the top is a plot showing a 4 log dynamic range with 6 peak beads. And you can see the brightest peak and the most dim peak are smashed up against the axis. And if this was actually our sample, we could not use this data. We can't get reliable data from this. What we would have to do is adjust the PMT voltages to get it within our detection window. So down here below that plot is a plot using the Novocyte. So you can see that there's a 7 log dynamic range on the bottom, and you can see that the brightest peak and the dimmest peak are all visible within this plot. So our window is much larger, so we really don't have to adjust our sample to fit within this window. So this is typically one of the most time-consuming steps in setting up a flow cytometry experiment. And if it's not done correctly, then you usually have to rerun the samples, or if you don't have any sample left, then you have to repeat that experiment. So this can really save you time and make it easier. But other than the PMT voltage adjustments, how does our wide dynamic range of detection really benefit the end user? So what I have here is a comparison between the Novasight flow cytometer and a competitor's instrument. And we took a blood sample, and we're just analyzing this forward scatter versus side scatter. So on the Novasight on the left-hand side, we have the lymphocytes that are down here at the bottom in red. We have a monocyte population, then a larger granulocyte population, and then finally the largest eosinophils. On the competitor's instrument, we have that same lymphocyte population down here in the lower left. Our monocytes and granulocytes are kind of grouped into this cluster here, and then we have our eosinophils here up at the top. And the problem with this is that the eosinophils are smashed up against the axis, so we can't really resolve them. If we wanted to resolve them on this type of system, we would have to turn the voltage on that detector down, but it would be at the expense of these lymphocytes that are much smaller. So with the Novasight, we can not only see dim and bright signals all in the same plot, we can see small and large particles all in the same plot as well. And so lastly, I want to touch base on a couple things that really make running experiments on the Novasight much easier, and that's a lot of the automatic features that we've incorporated not only into the hardware but also the software. So we have automatic compensation, automatic QC, fluidic maintenance and monitoring is all automatically done. Startup and shutdown is an automatic procedure. Same with rinse, wash, and debubble functions. So when we look at the software, so with our Novasight, we also have an acquisition and analysis software called Novo Express. This is kind of one of our toolbars in Novo Express, and you can see on this instrument tab, we have several fluidic maintenance functions. All you have to do is click on this icon and it will automatically perform this procedure. So we can easily do a cleaning, rinsing, extensive rinsing, back flushing, all with just the push of a button. So in addition to the fluidic maintenance, we have a lot of analysis functions that we provided to help make life easier. So we have automatic cell cycle analysis module, which will easily identify your G1, S, and G2M peaks. We have all the different plots that most other software platforms can use. So we have histogram overlays, we have the contour plots, we have automatic reports that are generated. It's really to make your guys' life much, much easier. So one thing I want to point out, which makes day-to-day life easy using the Novasight, is this QC test function. So it's a fully integrated function within the software to monitor the status of the cytometer over time. So it's as simple as clicking this button, running some QC beads, and you automatically get MFIs and CVs reported for you. And if you want to do the QC test report, you get these automatically generated Levy-Jennings plots where you can look at the instrument performance over time, so you can see if the instrument is performing as well as it was on the day of installation. So in this case, we have CVs and MFIs plotted here over time for one particular channel. So in terms of automation and making life easier, we also have this optional auto sampler. This can incorporate 96 well plates, 48 well plates, 24 well plates, even 24 tube racks that can incorporate fax tubes, Eppendorf tubes, bullet tubes, most platforms that are traditionally run with flow. So this sampler can automate a plate run, so you can click to run the plate. It will automatically run through your plate, and you can really walk away at this point. There's efficient mixing and washing steps that can also be customizable within this system. So in terms of workflow, I'm going to compare a traditional workflow versus the workflow that one would use with the Novasight, and to show you guys how easy it actually is and how much time it can save you. So startup. So right off the bat, normally you would start up the system, you would check the fluidic levels, and you would let the lasers warm up. So with the Novasight, the startup is a push of a button. All of our fluids are automatically monitored, so you don't have to check those. It automatically goes through a startup procedure where it cycles fluidics through the system, and the lasers that we use from Coherent take less than 5 minutes to warm up. So by the time the startup procedure is done, you're already ready to run samples. The QC test report, it could vary based on what kind of system you have, but with the Novasight, I already showed you that it's fully integrated into the system, and it's really easy to do it. It's automatically calculated for you. Then you would traditionally go to your experimental setup where you would adjust your PMT voltages so that either your positive or negative signal falls within your detection window, and then run your compensation. It's very similar with the Novasight, except one thing. We don't have to adjust those PMT voltages. We just can immediately we can immediately proceed to our single color compensation controls. And this can be done automatically or manually. So then we would acquire our sample, which can be done on the Novasite either using the single tube or the auto sampler. Our sample analysis, traditionally you would either wait till you're done running your samples to perform the analysis, or you would export that to a third-party software. With our acquisition and analysis software, you can even perform the analysis while samples are acquiring in the background. So I often am able to analyze my entire experiment by the time my last sample is finished acquiring. And lastly, shutdown. So traditionally you would cycle bleach and water, for 5 minutes to clean the system. With the Novasite, you just push the button. The Novasite performs its own 25-minute cleaning procedure, and then the power is automatically turned off. So I hope I've been able to explain to you that how the Novasite can really save you a lot of time in your acquisition and analysis. And here's just a list of all of the automatic features that we've incorporated into the system. So it's the startup and shutdown, the QC, the compensation, cell cycle analysis, automatic fluidic monitoring that just makes day-to-day life using the cytometer much, much easier. So with that, if you guys want any additional information, feel free to ask us after the talk, or you can always go to our website, aceabio.com, and we have a lot of additional application and technical specifications listed there on the website. So now I'm going to transfer this to Winnie Ludi from Enzo Life Sciences, and she's going to talk to us about a really cool RNA flow assay.